for the invitation. I'm gonna have click on this. I'm gonna have two disclaimers before I start. So this talk is gonna contain pictures of blood and mentions of blood. I learned my lesson recently because I was mentioning blood in an animal behavior class, and somebody in my class fainted. So I don't want to. I want to avoid that by just giving you that disclaimer right away. And also, there's a second disclaimer that this is not a math talk, right? So you won't see any equations in this talk, but I hope that some of the mentioned empirical examples that I'm going to talk about inspire some modeling ideas, right? So that was kind of the idea of this talk. So what does our lab do? We're kind of at the intersection of behavioral and disease ecology, where behavioral ecology is the study of the evolutionary basis for animal behavior due to ecological pressures. And disease ecology is the study of the mechanisms, patterns, and effects of host pathogen interactions. And I'd say our research is kind of like right there in the middle, trying to merge those two disciplines. And I'm going to talk about three projects. Some of them are ongoing, some of them are completed today. The first one's about social relationship infection feedbacks. The second part is about behavioral manipulation, specifically of rabies virus in vampire bats. And then the third part is like the most preliminary part, where we've just received funding to look at the fine scale behavioral interactions and cross species transmission between vampire bats and their livestock hosts. So let's start with the first part. And before I do this, I want to kind of talk a little bit about the history of epidemiological models to some extent in very base terms. But, most of you will notice that, the first epidemiological models are the early ones, and they're still useful, basically assumed that there were like homogeneous contacts between individuals in a population or group. And then with, the rise of social networks, we started to include heterogeneous contact structures. We know that like some individuals contribute more to overall transmission than others. And to add more complexity, we have started to think about those networks as being dynamic, right? Not just static, but they're dynamic. So if you think about like two time points here, we could think about like time point zero and then time point one, and we could include things like, maybe the time of the day will change network structure, or in this case, whether an individual is infected, right? So we know that if individuals infected, they change their behavior, and that can like rewire networks. And so just assuming that we have these heterogeneous context structures that are static, we can't really capture these dynamics. And so we know that infections affect social behaviors through host-driven behavioral changes. I argue there's not that much modeling on that. There's more now, but these changes can be driven by healthy conspecifics. So we have examples in people and in animals of exclusion, where infected individuals are excluded. Avoidance is very, very common, has been shown across most taxa at this point, where we're avoiding infected conspecifics, and we've all experienced that, right? If we go to a store and somebody sneezes next to us, we'll probably like step away from them. There's help, which is fairly common in human societies, but also in some eusocial insects, where infected individuals receive help actually, and that can actually increase contact with those individuals. And then we have really cool examples of proactive social distancing, which is something that happened, for instance, during COVID, where even though people weren't necessarily known to be infected or infected, we would still step away from each other because there was a risk of getting infected. And so there's some really cool examples in eusocial insects too, where this kind of proactive social distancing is happening. These behavioral changes can also be driven by potentially infectious individuals. And so in those cases, we have cases of active self-isolation, where individuals will actively self-isolate. So that is something that people do. That is something, again, that eusocial insects do. And then what we mainly work on is something we call passive self-isolation, where as a result of being sick, we change our behavior. And we're going to talk about those so-called sickness behaviors later on. But we're lethargic, we're like socially withdrawn, we're less likely to leave the house. And so it's not necessarily we're intentionally self-isolating, but it is more so a result of being sick, of our immune system being activated. And so that can definitely also reduce contact with others. And there's like some cool papers in epidemiology that show that, for instance, if people are infected with the flu, they're less likely to come in contact with others due to like those behavioral changes. And so to argue that there is now more so I think there's this additional layer of complexity that we talked about recently in this perspective piece where yes, we might have like these dynamic changes where like an individual becomes infected and is avoided by others or self-isolates, but this can also be dependent on the relationship that individual has with another individual. So, right, so if we have this simpler structure here where like time point zero, no changes, time point one, we have that individual infected, everybody avoids that individual loses all its contact. What we could imagine is that if another individual is infected that has like a strong tie with another individual, those contacts might increase. So what I'm saying here is that like the number of these or the nature of this behavioral change, avoidance, social withdrawal, whatsoever, is dependent on the pre-existing relationship that you have with another individual. And so that can lead to these feedbacks where the relationship determines the behavioral outcome or the behavioral outcome can affect the relationship. So that's something that we're thinking about right now. And so why do we study vampire bats? I think frankly they're pretty cute. They're fascinating animals. There are about 1,500 described bat species now, and only 3 of them feed on blood. And only this one, the common vampire bat, this Modus rotundus, is like a generalist. So they'll feed on, mostly livestock now, but they'll really feed on everything they can get. The other 2 species are bird specialists, so they'll be more likely to feed on birds. But a blood diet has come with like these unique and fascinating adaptations. So vampire bats are unique in the fact that they can basically walk on the ground very comfortably. They can hop. They use 2 different gaits, which is uncommon for bats, and we think they use that to basically sneak up on their prey. They have little infrared sensing receptors in their nose to find veins. And then they also they make a little incision, and then they have an anticoagulant in their saliva that basically prevents the blood from clotting, and allows them to feed on their prey, right? And so that anticoagulant has been explored in clinical trial as a stroke medication. But I think for me, the most fascinating adaptations to this blood diet are social ones. And so we know that vampire bats share food. We know that blood isn't very nutritious, and so vampire bats will starve relatively quickly. And so as a result of that, what we think as a result of that has evolved is this food sharing behavior where an individual that has fed will come back to the roost and will feed other individuals that haven't fed. And you see that here on the top, where we basically have the donor, which is the individual that sits still, lets the recipient like kind of lap out the blood, the regurgitated blood out of its mouth. And so this behavior is fascinating because when it was described earlier, but when it was first published, that was a Nature publication, I think in the 80s. It was shown that this happens between unrelated adults, which back then puzzled evolutionary biologists. Like a lot of the cooperative behaviors were explained by kin selection, where genetically it makes sense to help out kin, whereas like these cooperative behaviors between unrelated individuals were like harder to understand. And we now know that this is, a case of what we think is reciprocity, where these bats keep track of each other's investments. So they have like long-term social relationships with each other. They keep track of each other's investments. So if an individual doesn't share blood, then, the other individual won't share with it later on. So just like kind of human friendships where they keep track of those investments and they make their decisions based on that. And we also know that they use social grooming, which you can see here in this video, as kind of a low-cost social investment to maintain those relationships, even if there are no opportunities or need for food sharing. They kind of use these low-cost social investments to maintain their relationships. We also know that vampire bats form relationships by raising the stakes, which is very similar to how people think we form relationships. So when we form a friendship, we're likely not going up to somebody and investing a ton of money in them immediately, or like investing a ton of social currency in them. What we do is usually we test the waters, right? You might sit next to somebody, you'll talk to them, If they don't talk with you, you're probably like, this is not going to work out. If they talk to you, then you might start sharing your lunch whatsoever. If they share back, then that might escalate into higher cost investments in that friendship. And so vampire bats do the same. So my postdoc advisor back then did these experiments where he basically merged individuals that had never met each other and watched how they form relationships. And so they do a very similar thing where they start by grooming and then they kind of wait if the other individual grooms them back. And if they do, they'll groom a little more. And so they escalate those investments towards food sharing at some point, which is like a higher cost investment. And then the other thing is that we can simulate these behavioral symptoms of infections in vampires. So over the years, we have established this method that basically allows us to make them sick for like 24 hours, for lack of better words. But basically, we're using a bacterial compound. We inject them. We know that triggers the innate immune system, triggers the release of pro-inflammatory cytokines, which act in the periphery and in the brain, and basically they cause these sickness behaviors, what we call them, which are very familiar to us. When you're sick, you're feeling lethargic, you withdraw socially, there's increased sleep, decreased self-grooming, reduced vocalizations. And you can see here on this video, you can actually see one bat that was injected. You can see it is like lethargic, it doesn't really engage with the others. And so we can transiently basically change their behavior to what we think an infection would look like. And then the last plus about these vampire bats is that they basically fit in some, the space of a coat closet. So if we think a lot of like very socially complex species like primates or lions or hyenas or whatsoever, to work on them, we would need these large spaces. It's logistically extremely challenging. Whereas in vampire bats, really, this is where they spend most of their social lives, right? In like these tree holes that are about the size of your coat closet. And so there's many of them in there. We know from tracking studies that they'll spend like 20 to 23 hours of the day in there, interacting with each other. And then they go foraging for maybe 1 to 2, 3 hours. But really, we can really recreate in the lab these very, very small social environments that they usually interact with, that we couldn't necessarily do in other socially complexer species like primates, for instance. And so thinking about this feedback again, one thing that we wanted to look at first is whether the relationship actually determines the behavioral outcome of like these infection-induced behavioral changes. So again, as a reminder, we might have this setup where, yeah, in general, we would expect that if an individual is sick, it's reducing its contact with other individuals. But whether it's reducing its contact with other individuals might depend on the relationship it has with those individuals. And so what we looked at first, because I think it's the simplest thing to do, is kin relationships. So we know that vampire bats have these unrelated friends, in quotes, but maternal relationships are extra important. So especially moms and their daughters will stay together for most of their life. They will become each other's main food sharing partners, grooming partners. And so this is a grooming network from 1 hour studies. And you can see that the maternal grooming rate is usually higher than the non-maternal grooming rate. So these maternal relationships are very, very important. And so what we did in this study is basically we had a large colony of vampire bats. We pulled bats and injected them either with LPS, which is the bacterial compound I was talking about earlier, or PBS, which is just a control. You can see here control and LPS. And each bat was injected with both treatments with like a time period in between in randomized order. And basically, if a data point is over here, something happened more after the control injection. If a data point is over here, something more happened after the LPS injection, after they were injected with the immune challenge. And if you look at non-maternal grooming, for instance, between sick moms and non-offspring, you can see it usually happens more, if, later on, if that mom's sick, happens more after the control, right? So what this basically means is that after LPS injections, if your data point is over here, you have reduced grooming. And so sick mom to non-offspring, sick bat to non-close kin, sick offspring to non-mom, they're all kind of like over here. Whereas if you look at maternal grooming, it is mostly maintained, right? So in maternal grooming, especially for moms to sick offspring, it almost seems to happen more after that offspring is immune challenged, right? So again, I think this kind of illustrates that like the type of relationship that you have with with an individual might determine like the behavioral outcome when you're infected with something, and you'd usually change your behavior, but again, that might depend on that specific relationship. And so we did this with kin relationships, and our future work will explore this across different relationship types. And so in vampire bats, again, we're talking about those non-kin relationship types, like these friendships. That we want to test, which are kind of harder to quantify. So kinship is fairly easy to quantify. You get your pedigree or your genetic relatedness in your colony, and so you're like you're related or you're not. You're somebody's daughter or you're not. Whereas like kind of quantifying these non-kin relationships is a little harder because we have to watch them beforehand, figure out their social networks, figure out their social partners before we can actually do the experiment. So that is some ongoing work. Um, the second thing that we think about in this perspective piece is that the behavioral outcomes themselves might affect relationships, right? So if you're, for instance, in crucial stages of relationship formation, like I talked earlier about when you're about to form a friendship with somebody, but they're sick or you're sick and they avoid you, that can probably like hamper or slow down relationship progress, right? And so what we talk about in this perspective piece too is like basically the reverse feedback where the behavioral outcome might affect the relationship. And so we had this opportunistic, this opportunity to study this because when we did a merger, which means we merged bats from different populations to see how they form relationships, what happened is we had an outbreak of these skin infections that caused like some lasting effects, that caused some sickness behaviors. We treated them. But basically, what we had throughout this entire process was data on their allo-grooming interactions. And what we find in this paper that's currently in review is that infections had a greater impact on the formation of new relationships than on already existing relationships. So that's again something where yes, if you're sick, it might affect your new relationships more so than it affect your established relationships, which is interesting. And so in this specific case, for instance, we had a behavioral outcome, which was the staph infection, which caused behavioral symptoms. So those individuals were lethargic, they groomed others less, and that basically slowed their relationship formation with partners that they met during that time when they merged with other individuals from other populations. The second thing I want to talk about is behavioral manipulation. And so this is something that some of you might be familiar with if you've watched it's pretty common in popular media. If you watch The Last of Us, cordyceps manipulating people. Cordyceps is a very well-known behavioral manipulator of ant behavior. There are many of these parasites that do seemingly manipulate host behavior, to some extent also social behaviors. And so, again, if we go back to our conceptual framework, we talked about host-driven behaviors quite a bit. We talked about these sickness behaviors. We talked about avoidance. So in this case, we actually have pathogen-driven behavioral responses that might increase your contacts with others, right? So if a pathogen, if a parasite manipulates you into more social interactions, then we'll have changes in those social networks as well. And so one pathogen that we work on is rabies, and rabies is kind of like a notorious, what we think, behavioral manipulator. And to understand how rabies kind of manipulates the behavior of its host, we have to think about its replication cycle. So usually the virus enters the body through a bite and then accumulates in the muscle. So this is my plea to you that when you get bit by an animal, you should go and get rabies post-exposure vaccines relatively quickly, because as it is in the muscle here, it is still accessible for the immune system. And so that is when you can intervene. And then if you know anything about rabies, what we consider rabies to be almost 100% fatal. There's, I think, 1 or 2 people that have survived it without like lasting neurological damage. But once it gets into the central nervous system, so after it replicates at the neuromuscular junction and ascends into the spinal cord and the brain, it's really hard for the immune system to access. At later stages of the infection, it spreads along nerves to the salivary glands. And then we have these usually two behavioral outcomes where we have paralytic rabies, where infected individuals fall into a coma. And so this is true for animals and people. And we have, what is maybe more notorious, which is furious rabies, which causes, fear of water, heightened aggression, and so on. And we would think that this behavioral change right here actually furthers the transmission of rabies, right? Because it needs to get into another wound to some extent. And so why do we study this in vampire bats? Vampire bats are considered a reservoir for rabies virus. Vampire bats transmit rabies to each other, livestock, and humans. And we think that transmission probably relies on social behaviors in vampire bats too, but how rabies actually affects behavior of infected vampire bats was previously unstudied. There was like some anecdotal notes, and I'm going to talk about that too. But we were actually able to do like an empirical test of this with Dr. Elsa Canales, who is a veterinarian. And so during her PhD, she studied transferable vaccines in vampire bats. So what Elsa did is she brought bats, which is now mostly impossible, but she brought bats from Mexico to Wisconsin to do experiments on them. And so what she's interested in is these transferable vaccine, where you put a vaccine on their fur and it'll go back to their roost, and other individuals will kind of like groom it off of them and get like indirectly vaccinated against rabies. And so she got these bats. She used 3 different treatments. One of them was a placebo treatment. One of them was the transferable vaccine. So it's basically the vaccine on the fur. And the third one was just an oral vaccine. And then usually what people do to test whether vaccines are efficient or not in like these animal studies is they expose those individuals to rabies. So we had 40 male vampire bats in 3 groups that were exposed to rabies. And then after 34 days, 35 days, she looked at their brains and confirmed whether they infected with rabies or not. Now, again, she was interested in like whether that initial vaccination here determined whether they were later on rabid or not after exposure. But we got the opportunity to sample their behavior during that time. So we basically filmed those bats in those 3 groups, 3 hours per night. We did scan sampling, and when I say we, this was mostly this was an undergrad project by Eleanor Cronin, who's now a grad student in Florida. And she and others scan sampled those behaviors every 5 seconds and looked for allogrooming again and for aggression, right? So we're kind of interested in like furious versus paralytic. And in total, we had about 18,000 behavioral samples from this experiment. And what we found is that rabid vampire bats seem to groom others less. So what you can see here in this figure is these 3 groups, each bat, each night. Basically, each night is a dot. And you can see the bats that later on tested for rabies. Incidentally, like this experimental design was kind of nice to keep us blind too, because we didn't actually know who would test rabies positive at the very end of the experiment. And so you can see here that these rabbit bat groomed others less, especially, like 12 days after they were exposed to about 17 days. And then, a lot of them actually died at kind of like that 15, 16, 70-day mark of rabies. Some of them survived throughout the entire experiment. But we do see like this drop in grooming, right? So we don't see like an increase in grooming. Interestingly, we looked also at aggression, which is maybe more notorious for rabies. And so there are 2 types of aggression. You can have like these smaller standoffs. You can see here, these 2 bats have kind of like this smaller standoff where they try and bite each other. And we figured out who did the biting and how what was the directionality of it. And then sometimes you also have like these full-on brawls where they'll get into these fights. So level of aggression varies quite a bit from these bites to full-on fighting. Interestingly, what we find is that there was not really a difference between rabbit and bats that turned out to be non-rabbit at the end of the study. So not a lot of aggression at all. So rabbit vampire bats did not seem to be more aggressive. And so that was kind of interesting to us because we were like, well, rabies we would really think relies on like some of these aggressive interactions to get into new hosts, right? For transmission. And so we did like a quick literature search. It was quick because there were really only 10 studies that had either experimentally infected vampire bats with rabies, mostly for these vaccine trials, or observed rabid vampire bats in the field. And we found that 7 of these studies didn't show or didn't observe any aggression, including our study. And 3 of those studies observed aggression. Now, when I say observed aggression or didn't observe any aggression, what I'm usually talking about is one sentence in each of those papers. So none of those papers actually quantified behavioral changes over time like we did. Most of them just had a mention in the paper that's basically like throughout the entire study, we didn't see any aggression, or like during the study or doing the observations, it seemed like the bat were more aggressive to others. So that's kind of like a caveat here. We are kind of like the only study that quantified it over time. But what was interesting is that the 7 studies that showed no aggression, especially in those using those experimental infections, use strains from different animals. So we used a strain from coyote actually in our experiments, different time points and regions. Whereas like the 3 studies that observed aggression actually looked at naturally circulating strains, and so we're like in those specific population at the specific time. And so what we think might be going on is some sort of local adaptation where rabies viruses might be locally adapted to their hosts. And we're really interested in testing this in the future. So we know from studies in mice that the rabies glycoprotein seems to be important in those behavioral changes when it interacts with NACH receptors. And so the idea at some point is to challenge vampire bats with rabies glycoproteins from different animals, from different areas whatsoever, to see how they change their behaviors. And so we've started thinking about this specifically with Dr. Angeles Sales, who does like bat peptide injections in the brain and is interested in bat behavior. So that might be something that we're pursuing in the future. But I also think this is interesting because I do think pathogen manipulation of host social behavior, including both fitness consequences for both host and pathogen, I'd say is under-modeled. There's not that much modeling, especially when we think about like heterogeneous contexts and like social networks. I think there is like kind of an avenue for future work there too. And then the last or the third part I want to talk about is this study that we got funded relatively recently which thinks about fine scale behavioral interactions and cross species transmission. And so there's like some modeling in there. We have a collaborator who's going to do a lot of the modeling, both statistical modelings and modeling and simulation modeling. But really, I'm going to talk about like the background and more the empirical framework of this. So this just started like a few months ago. We're just hiring people for this. But basically, we know that vampire bats, cattle, and humans live in this complex host pathogen community. And what I'm talking about is like this complex ecological network where you have obviously vampire bats interacting with livestock, right? We know vampire bats will bite them. Livestock in most of those areas is their preferred prey now. Really, when we think about vampire bats before livestock arrived in those areas, they would probably feed on native animals like tapirs and peccaries, but livestock has really changed the game for them. It's a very reliable food source. It doesn't move much. Cows have a lot of blood. So really, if we go into these areas, their main diet now is livestock. We know livestock interacts with other livestock interacts with people. We know vampire bats will bite people, so that's not something that doesn't happen. And then vampire bats will interact with vampire bats and also with other bats in their roost. And so you have like this intertwined network of different species interacting with each other. And we know that in that network, we know, rabies is circulating. We know vampire bats transmit rabies to livestock, and I'm going to talk about that in a second too. We know vampire bats have herpes viruses, which are usually a little more host specific. We know vampire bats have Bartonella, which we know has the potential for cross-species transmission, coronaviruses, mycoplasmas, and more recently, people also have found like trypanosomes in vampire bats alive, and so we don't know if that actually jumps between species too, right, as they bite. And so we have this complex interconnected host community. And we work in three areas. So we work in Panama. We have like multiple study locations in Panama, and you can see here some of these study locations. We work in a newer study location in Costa Rica, which is this forest reserve run by a local university and the University of Michigan. So there's a lot of work on capuchin monkeys going on there, but we know we went there, we mapped out the vampire bat roost, so you can see the vampire bat roost on this. We found them basically with radio tracking and just hiking through the forest and finding those roosts. And so we know the vampire bat roosts, and we know the cattle basically distribution in this forest reserve. And so this is kind of nice because it's isolated. So there's like some, I think it's sugar cane around it, but we really think that those vampire bats feed on those cattle. And then we also have some study areas in Belize. And I think the main gap is that studies that think about cross-species transmission often look at molecular determinants of pathogen sharing, right? So you go out and you sample a bunch of different species. This happened, for instance, in the context of COVID. And you're thinking about like, do they share certain pathogens? And in some cases, you look at sequence similarity, right? But it quite often misses the behavioral context, which is like, how do these species actually interact with each other? And so what the main goal of this project is to couple contact networks with pathogen sharing networks. So again, we all have these pathogen sharing networks where we go into these study areas. We try and repeatedly sample both the bats and the cows. We do diagnostics for trypanosomes, viruses, bacteria, and see what do they share with each other. And then we are also going to get these contact networks, and I'm going to talk about how we do that in a second. And so the main questions we're interested in is like what is actually transmitted. So we know rabies is a big one, and that has been studied quite a bit. But like, what about other things like trypanosomes, Bartonella? What are transmission relevant contacts between species? What are kind of like the key individuals in those interactions? And then also thinking about control strategies. And when I talk about modeling, so this is mainly Megan Graft, who's going to do that, the University of Minnesota, who is like a wildlife disease modeler using social networks mostly, social network analysis mostly in modeling, is going to do simulation modeling. So we're going to use these networks to simulate generalizable epidemiological processes. And then, this will also include some statistical modeling, trying to understand how connectedness predicts pathogen sharing, and if not, why. And so we've gone there, we're doing observations. What you can see here in this video is vampire bats on a cow. To do these kind of observations, you basically have to let the cows habituate to you for a second, and then you can watch them at night. But you can see here this blood, for instance, has blood running down its neck. So that cow was bit before, and you see vampire bats on the cow. And then you see this weird observation. So this is what it looks like at night at those study areas where vampire bats feed in cattle. So this is an observation from Panama. We have done GPS tracking where we actually put these little GPS trackers on vampire bats to figure out where do they feed, right? So this is a cool example where we tapped 2 or 3 vampire bats, and for at least 2 of them, we got like this repeatable pattern where their roost was over here, and at least 2 of them went to the same location, or 2 of them went to the same location twice in consecutive nights. And then one of them actually kept coming back in the third night, and we think there's another roost down here somewhere. But basically, it seems like that, once they find cattle, which makes sense, once they find cattle, they'll keep going back to those locations. So that's something to keep in mind later when we talk about how we could potentially control or mitigate this vampire bat livestock conflict. And then what we use now are these proximity sensors, which are relatively new technology. But basically, what we do is we put these proximity sensors on vampire bats. These proximity sensors can send out like these beacon signals we can adjust it, but the lowest resolution is the highest resolution is every 2 seconds. So we can basically get networks every 2 seconds, which is like, an incredible data yield. But basically, though, in this case, they'll send out these beacon signals every 2 seconds. If another proximity sensor, like from a cow, is in range, which is about like 10 meters or so, those sensors will start interacting with each other. They'll exchange data. They'll store meeting duration, and they'll store signal strength, which is kind of a proxy for distance. And so that lets us build these like networks between vampire bats and cows, and between vampire bats and vampire bats. And so really high resolution networks, dynamic networks over time. And we have very, very preliminary data on this. So we did this a few years ago, where we just, I think we tacked like 3 or 4 cows and like 3 or 4 vampire bats at a study location, just like as a proof of concept. And it's not much of a network, especially the second observation here on night 2. But what we can see is some interesting patterns where, yes, we can detect these contacts using proximity sensors. So you can see them here mounted on a bat, and then in cows they can be a little bigger around their neck. And you can see here that bat A9 here basically observed or visited a cow 2 consecutive nights, the same cow 2 consecutive nights. So there's already some interesting data in there, but obviously we want to scale this up across more individuals, more cows over longer periods of time. And so why do I think it's kind of interesting to understand these cross species transmission dynamics and important in this species? Well, we know that there is a conflict, just this vampire bat livestock conflict going on. And the main reason for that is that vampire bats do transmit rabies to livestock. And so they call this what's locally called dengue, which is paralytic rabies. It's mostly fatal in infected cattle. There's some evidence of maybe abortive infections where cattle get infected, but maybe their immune system catches it before they show symptoms. And so you'll find cattle that are seropositive, that'll have antibodies, but they actually never got rabies or never experienced any symptoms. So that does happen. But really, it does cause it has an economic impact. So it's estimated, there's like economic modeling too, that rabies caused cattle deaths are a major economic impact in Latin America, tens of millions of dollars. And so we know that rabies is maintained within vampire bat populations, and we know that culling often fails. So the types of culling we see is if people find a roost, they'll maybe set fire to it. So there'll be like kind of localized culling of roosts. Then there's also this more, what's used by local agencies quite often is this vampiricide, which is basically just a mix of petroleum jelly and rat poison. And so it's supposedly it works like that transferable vaccine, where you catch a bat around livestock, you put that poison on its fur, and then it goes back to the roost, and others kind of like uptake it by grooming that individual. We know that culling quite often fails, actually. In some cases, it might actually increase rabies transmission. The reasons for that are kind of cryptic still. We think maybe that if you cull, that remaining individuals disperse, and that might have an effect. One of my students, so the student I was talking about earlier who joined my lab, is actually a modeler and has done modeling on transferable vaccine. Is really interested in like how culling. So she's going to do some modeling work on how culling disrupts networks, and how they might be rewired, and how that then might affect disease dynamics. So she's interested in looking at that from like a modeling perspective. She's working on that right now. But yeah, in many cases, it seems like culling is not very effective in preventing those rabies outbreaks. Vaccinating cattle is an option. So, the pros are that it protects cattle from almost always fatal disease. It's ethical. It's not super disruptive. It's an alternative or maybe a complement to like culling. The cons is that it requires ongoing costs, right? It needs a cold chain. So the vaccine has to be chilled, and labor. And it's hard to implement in remote areas, right? And it also needs repeated vaccination quite often. So, it lapses. If you have rabies vaccinations, if you work on bats, you have to get your titer checked every year just to make sure that your titer is still up. If it's not, you have to get a booster. And so again, this is not like a one-time thing. So these cattle have to be repeatedly vaccinated, which again kind of poses this logistical challenge. More recently, people have thought about like vaccinating vampire bats, right? And so I talked about the transferable vaccine, which is kind of shown here, where the coverage basically increases passively. It's hard to control how far the vaccine actually spreads. and which animal receives it. But it's very similar to culling. You grab an individual, you put the vaccine on its fur, it goes back to the roost, and other individuals might ingest it and get vaccinated basically passively from this. And then the second method that people consider, specifically like Daniel Stryker's lab at the University of Glasgow, is these transmissible vaccines. And that's not just in vampire bats. People have thought about this in other wildlife disease systems. But basically, we know, for instance, that vampire bats have these herpes viruses. And so the idea is to make genetically modified herpes viruses that express rabies proteins. And so you have these naturally circulating herpes viruses that immunize vampire bats against rabies, right? So that's kind of like the idea. There's potentially really high coverage, but there are like some ecological and ethical risks, like thinking about viral evolution. And so there's some modeling work on this and some perspective pieces if you're interested in this from other labs. But yeah, the idea is that you have vampire bats being vaccinated against rabies using another rabies virus that is basically circulating in those populations. And yeah, that might be some ethical risks too, right? Of introducing something genetically modified. But yeah, there are definitely experts on this that are working on this. And so I think it's an exciting new avenue, but we're not there yet, right? So this is obviously in development. But until then, like I think, some of the cheapest interventions could be based on vampire bat behavior. So what we're trying to figure out with this study is basically just the contact patterns between vampire bats and livestock and whether there's ways to basically disrupt those, right? So on top of doing the tracking, what we're interested in is to see is what does cattle movement do, right? So earlier we saw in this GPS tracking study, we saw that these vampire bats go back repeatedly to the same pasture because they know that the cows will be there on consecutive nights. So how does moving the cattle, if you have the space, how does moving the cattle across different pastures basically affect those dynamics? We're thinking about trying out light deterrents. We know vampire bats are lunarphobic, meaning that they don't like to forage usually they like to forage usually in the darkest hours of the night. They don't like to forage at like full moon, probably to avoid predation by owls and such. So we're thinking about light deterrents, and then we're also thinking about acoustic deterrents. And so as a layer on top of what we're already thinking about in terms of tracking vampire bats and livestock contacts, we're also interested in like thinking about those behavioral interventions that are maybe a little cheaper and can kind of reduce like vampire bat attack rates on cattle. So with this, I know I'm a little early, so we have plenty of time for questions, but I want to say thank you to NSF for funding this research or ongoing research, STRI, a whole bunch of field techs and collaborators, and then obviously the vampire bats and the cows and the local agencies that help us out. So thank you. Let me know if you have any questions. If you don't get to ask your questions, but it is still burning at the end of the time, feel free to cold email me. I'm just, I don't know, how far is Chattanooga from Knoxville? 2 hours away. Feel free to cold email me. My email is down here. Yeah, and so